My name is Dr. Joan Han. I'm a pediatrician at the Le Bonheur Children's Hospital and the University of Tennessee Health Science Center. My colleagues at the National Institutes of Health and I have been studying patients with Weger syndrome to better understand the role of brain-derived neurotrophic factor, BDNF, in human pain perception. Weger syndrome is a very rare disorder. It occurs in approximately 1 out of every 1 million individuals. And the cardinal symptoms of Weger syndrome make up its acronym, WAGR: Wilms tumor, aniridia, genitourinary anomalies, and a range of developmental delays. The cause of Weger syndrome is contiguous 11p13 region gene deletions, and the size of the deletions can vary from as small as about 500,000 base pairs to over 30 million base pairs. There's variable involvement of genes in the region. The key genes are WT1, which causes the Wilms tumor and GU anomalies, and PAX6, which causes aniridia. In this current study, we became interested in looking at the role of BDNF in nociception. And what prompted this interest is that we heard very dramatic stories from the parents of some of the children with Weger syndrome. For example, my child had a leg injury, but kept walking on that leg until it was purple and swollen, and we finally took him in to get it checked, and the x-ray showed a significant fracture, but he was only complaining of minimal discomfort. Another example, the child had a ruptured eardrum, and there was pus and blood draining from the ear canal before the parents even noticed, because again, the child was complaining of minimal symptoms. I think the one that struck me the most, though, was when impaired nociception actually was potentially life-threatening. In this case, it was a child who developed pancreatitis, which typically causes excruciating abdominal pain. But in this child, there was only minimal symptoms, and so there was delay in her receiving medical attention until she was so sick that she ended up in the intensive care unit. The other thing that was remarkable was that it's not just the nature of these stories, but the fact that it was some and not all of the patients with Weger syndrome who had these types of symptoms. And this caused us to hypothesize that there may be a critical gene within the 11p13 region that is responsible for this impaired nociception. The candidate gene that we selected was BDNF, and this is based on prior data that has been shown in rodents, suggesting that BDNF has an important role in pain perception. We therefore used Weger syndrome as a human model, a natural knockout of BDNF, to understand what is BDNF's role in humans. Figure 1 summarizes the results of the clinical patient data. In Figure 1C, we show the responses of 32 patients with Weger syndrome. Their parents completed questionnaires regarding their responses to typically painful conditions. And we observed that patients with heterozygous loss of BDNF had significantly higher reports of pain insensitivity. Next, we wanted to do a quantitative sensory test. In this test, we use the Medoc Thermal Sensory Analyzer, and a thermometer is placed, and they are asked to rate hot and cold temperatures using a 6-point Wong-Baker Faces pain rating scale. There were 12 patients who were able to do this. In Figure 1A, it shows the deletion boundaries for the 6 patients with intact BDNF and the 6 patients with heterozygous loss of BDNF. Figure 1B shows that these two groups were well matched for age, sex, intellectual ability, Wilms tumor, and past exposure to the chemotherapeutic agent vincristine. We also conducted nerve conduction studies in these patients, and that is shown in supplementary table 1 and 2, and observed that in the patients with and without BDNF deletion, that both motor and sensory nerve conduction studies were normal and not different from each other based on group. In Figures 1D and E, you can see that the patients with BDNF loss reported at both hot and cold temperatures lower experience of pain compared to those with intact BDNF. Our clinical findings in patients with Weger syndrome support a role for BDNF in pain perception. To investigate these associations further, we conducted bioinformatics, gene expression, and rat experiments.
The implication of BDNF in these patients was preliminary for a number of reasons. Weger syndrome is a complex genetic disease. As we show in Figure 2A, there are at least 12 genes that could potentially explain the pain phenotype that we observed in Weger subjects. Using a bioinformatic analysis, we aim to reduce the list of candidate genes by screening those genes that showed enriched expression in either the dorsal root ganglion or the dorsal spinal cord, two tissues where painful sensations are transmitted into neural impulses and carried to the brain. We hypothesized that a pain insensitivity phenotype would most likely manifest in one of these two tissues. As shown in Figure 2b, BDNF was the most highly enriched gene in these two tissues, although not the only gene expressed. To specifically examine the effects of BDNF hemideletion in isolation, we used a heterozygous BDNF knockout rat, which, like the Wagner subjects, has a disruption of one of its two copies of BDNF. These studies examined thermal stimuli similar to that used in the Wagner subjects. In Figure 3, we saw that these animals took longer to respond to hot and to cold stimuli. At the same time, we measured the temperature at which they chose to terminate the stimulus, and found that these animals required more noxious stimulation in order to evoke a response. These results made us confident that BDNF was sufficient to explain the reduction in pain sensitivity observed in Wagner syndrome, and justified additional studies using next-generation sequencing to examine the molecular changes that occur in the dorsoroot ganglion and in the spinal cord that could explain how this occurs. These experiments were complex, but one of the take-home messages was that we now know that the signaling between neurons and most likely non-neural cells, such as glia, is disrupted in the BDNF knockout rats. We saw signatures of alterations in immune-like glial pathways, which most likely indicates the involvement of these non-neural glial cells in the changes that are responsible for the pain phenotype. These combined investigations in rats and in humans show that loss of one copy of the BDNF gene is sufficient to explain the pain phenotype in Wagner syndrome. Unlike other pain and sensitivity phenotypes that have been described, this does not appear to be a defect in registering the painful events, but rather in processing them. We now know that the pain-sensing neurons are not lost or damaged by this genetic mutation, and this implicates a new and potentially very exciting mechanism that could help us learn more about pain and pain control.
This investigation is a good example of a collaborative effort between clinicians and basic scientists. Through this work, we were able to understand what's going on in the humans and test them in a unique patient population, and then take some of the observations that were made in the human and transfer them into a rat to specifically understand what was going on. And it's through these investigations that we learned something new about the BDNF gene. And hopefully we will take what we've learned and potentially convert it into new therapies, because we now understand a new communication pathway in the CNS, the central nervous system.